The practice of biohacking is rapidly transitioning from niche, do-it-yourself experimentation into a rigorously data-driven precision health framework. The gut microbiome functions as a highly responsive, programmable interface between host genetics and environmental inputs. Contemporary research approaches the microbiome as a bidirectional communication network, rather than a passive recipient of nutrition. This closed-loop precision health model integrates three nodes: the host, the microbial ecosystem, and a digital data stream. We manipulate this loop through dietary modulation, biotherapeutics, and engineered strains. Multi-omics monitoring tracks these outcomes, feeding physiological data back to complete the cycle. Safely engineering this system requires mapping the specific molecular pathways that link the gut to the brain. To locate the origin of this communication, we look to the microscopic environment of the gut epithelium. Within this environment, bacteria produce short-chain fatty acids, or SCFAs. These molecules, specifically butyrate and propionate, act as the primary messengers for host signaling. This cross-section shows the mechanism of receptor binding. Geometric SCFA molecules couple with specific G-protein-coupled receptors, GPR41 and 43, located on the host cell wall. This binding modulates cytokine networks and promotes the assembly of tight junctions, strengthening the physical barrier of the gut lining. On a deeper level, butyrate acts as a histone deacetylase inhibitor. It penetrates the cell to block the HDAC enzyme, which causes host DNA to unspool. This epigenetic shift promotes the differentiation of regulatory T cells, directly altering the host's immune tolerance. Secondary mediators provide further layers of control. Bile acid derivatives interact with host FXR receptors, while tryptophan catabolites activate the aryl hydrocarbon receptor to maintain epithelial defense. These microbial byproducts function as molecular drivers that actively rewrite host genetic expression and immune status. SCFA and metabolite signaling enters systemic circulation, acting as endocrine-like mediators throughout the body. In the metabolic axis, acetate travels to the liver to regulate hepatic gluconeogenesis. This map traces the link between the gut and liver. Because of this connection, the rate of microbial carbohydrate metabolism serves as a mechanical driver of host insulin resistance. A second pathway identifies the neural axis, the microbiome-gut-brain axis. Gut-derived neuroactive signals, including GABA and serotonin precursors, travel up the vagus nerve to interact with the central nervous system. These inputs allow the gut to modulate cognitive function, synaptic neuroplasticity, and mood. Diurnal oscillations in these networks are synchronized via clock and BMAL1 genes, linking microbial activity to the host's circadian rhythms. High-level cognitive performance and metabolic health emerge as measurable outputs of a rhythmically synchronized gut ecosystem. To program this interface, we must identify the specific levers used to control these pathways. This three-tiered architectural stack categorizes the interventions used to manipulate the microbiome. Tier 1 is dietary modulation. Manipulating microbiota-accessible carbohydrates selectively increases the production of beneficial SCFAs. Tier 2 involves microbial therapeutics. Prebiotics, postbiotics, and fecal microbiota transplantation are used to restore diversity to the gut ecosystem. Tier 3 utilizes synthetic biology, moving from ecological restoration into genetic engineering. Rational design and CRISPR technology allow for the programming of specific probiotic strains. These engineered strains serve as real-time biosensors or delivery systems for mucosal immunization. While Tier 1 is accessible through lifestyle, Tier 3 treats microbes as programmable hardware. Precision interventions require high-resolution analytical feedback to function effectively. The current methodological bottleneck in the field is the severe heterogeneity in how microbial data is sequenced and analyzed. This split-screen comparison shows the fidelity gap between legacy sequencing and modern precision requirements. 16S rRNA gene profiling is affordable, but lacks the taxonomic depth to differentiate between closely related bacterial strains. Shotgun metagenomics and metatranscriptomics identify both the taxonomic identity of the bacteria and their specific metabolic functions. Precise modeling relies on multi-omics integration, layering metabolomic and proteomic readouts over the genomic data. This integration identifies cross-omic modules that accurately predict therapeutic outcomes where loose correlations fail. Biohacking attempted without this multi-omic validation remains speculative. Returning to the completed closed-loop model, we see a fully integrated biological architecture. Lifestyle inputs alter microbial functions, triggering epigenetic responses that are monitored by AI-driven health twins to personalize the next intervention. Clinical translation of this loop faces significant risks if it remains unregulated. Unregulated DIY FMT presents pathogen risks, while engineered probiotics carry the danger of uncontrolled horizontal gene transfer. Integrating biological data with digital platforms requires strict cyber biosecurity to prevent the manipulation or piracy of genomic information. Overcoming these risks requires standardized reporting, specifically the adoption of the STORMS guidelines. The deployment of live biotherapeutics must be preceded by strain-level safety genomics. Causal inference must be established through longitudinal multi-omics rather than isolated snapshots. The human holobiont is an astonishingly programmable system, yet realizing its engineering potential demands an uncompromising, evidence-based methodological framework.